Genetic engineering. It sounds like something out of a science fiction novel, doesn't it? But in reality, it's a field that's rapidly changing the world around us. In the simplest terms, genetic engineering is the direct manipulation of an organism's genes to alter its characteristics in a particular way. Imagine being able to edit the building blocks of life itself. That's the power of genetic engineering. The story of genetic engineering begins long before we even knew what DNA was. For millennia, humans have been manipulating genes indirectly through selective breeding. Farmers would select and breed crops with desirable traits, like larger fruits or higher yields, unknowingly shaping the genetic makeup of their harvests. This process, however, was slow and imprecise, relying on natural variations and chance mutations. The discovery of DNA's double helix structure in 1953 by James Watson and Francis Crick revolutionized biology and opened the door to a new era of genetic manipulation. Scientists realized that DNA held the blueprint of life, and altering its code could lead to profound changes in organisms. The first recombinant DNA molecule was created in 1972 by Paul Berg, marking a pivotal moment in genetic engineering history. This breakthrough, combining DNA from different sources, paved the way for groundbreaking applications like the production of human insulin in bacteria, revolutionizing diabetes treatment. This early period of genetic engineering relied on relatively crude tools, often involving the transfer of entire genes or large DNA fragments. While groundbreaking, these methods were akin to using a sledgehammer for delicate surgery, lacking the precision and finesse needed to truly unlock the full potential of genetic engineering. The true revolution in genetic engineering arrived with the development of CRISPR-Cas9, a revolutionary gene editing tool that has been rightfully dubbed genetic scissors. CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, acts as a programmable search and snip tool for DNA. Imagine you have a sentence with a spelling error you want to correct. CRISPR acts like a word processor's find and replace function, but for DNA. It uses a guide RNA molecule, like a specific address, to locate the target gene sequence within the vast expanse of an organism's genome. Once the target sequence is found, the Cas9 enzyme, which acts like a pair of molecular scissors, makes a precise cut at the desired location. This break in the DNA strand triggers the cell's natural repair mechanisms, which can be harnessed to either disrupt the target gene or introduce specific changes with remarkable accuracy. Before CRISPR, genetic engineering was a time-consuming, expensive, and often inefficient process. CRISPR revolutionized the field by making gene editing faster, cheaper, and more accessible than ever before. This breakthrough democratized access to this powerful technology, sparking a wave of innovation and research across various fields. The ability to precisely edit the blueprints of life comes with immense potential for good and a responsibility to carefully consider the ethical implications. Genetic engineering holds the promise of revolutionizing healthcare, agriculture, and even our environment, but it also raises concerns about unintended consequences and the potential for misuse. In medicine, genetic engineering offers hope for treating and even curing genetic diseases that were once considered incurable. Gene therapy, for example, uses modified viruses to deliver functional genes into cells, potentially correcting genetic defects and restoring normal function. Scientists are exploring CRISPR-based therapies for diseases like cystic fibrosis, sickle cell anemia, and Huntington's disease, offering hope for effective treatments and possibly even cures. Beyond treating diseases, genetic engineering has the potential to revolutionize agriculture. Crops can be engineered to be more resistant to pests, diseases, and harsh environmental conditions, potentially increasing yields and reducing reliance on pesticides. This technology could play a crucial role in ensuring food security for a growing global population facing the challenges of climate change. However, the power of genetic engineering also comes with potential risks. Unintended consequences of gene editing, such as off-target edits or unexpected interactions within complex biological systems, could have unforeseen and potentially harmful effects. The ethical implications of genetic engineering, especially when it comes to human germline editing, are profound. Altering the genes of future generations raises concerns about designer babies, genetic inequality, and the very definition of what it means to be human. These are complex issues that require careful consideration, open public dialogue, and robust ethical guidelines to ensure responsible development and application of this powerful technology. Genetic engineering is no longer a futuristic fantasy. It's a reality that's rapidly shaping the world around us. From disease-resistant crops to gene therapies for genetic disorders, the applications of this technology are already making a tangible impact. In agriculture, genetically modified GM crops have become increasingly common. These crops, engineered for traits like herbicide tolerance, insect resistance, and improved nutritional content, have the potential to increase yields, reduce pesticide use, and enhance the sustainability of food production. In medicine, gene therapies are being developed for a wide range of diseases. Clinical trials are underway for therapies targeting inherited blindness, spinal muscular atrophy, and certain types of cancer, offering hope for patients who previously had limited treatment options. The future of genetic engineering holds even greater promise. As our understanding of genetics and our ability to manipulate DNA continue to advance, we can expect to see even more groundbreaking applications emerge. Imagine a world where genetic diseases are a thing of the past, where crops can thrive in even the harshest environments, and where we can engineer solutions to some of the world's most pressing challenges. This is the future that genetic engineering has the potential to unlock, but it's a future that we must approach with caution, responsibility, and a commitment to ethical considerations.